We now have what I hope is going to be a very interesting and exciting session with swapping between clinical talks and also hearing about some patient experiences, which is always really very inspiring. However, our first speaker is Professor Adam Mead, and Adam has worked both obviously in clinical trials, but also on the science of the MPNs for some years now in Oxford. And he is really good at explaining the science and giving updates. So, Adam, over to you for a scientific update.
Terrific. Thank you very much, Mary Frances and Nona. It's a real pleasure to be here and see so many familiar faces. I was having a very nice dinner and chat with Nona last night, and I asked her whether she thought it would be interesting if I talked a little bit at the beginning of my talk about scientific updates, just about being a clinician scientist, what it involves, how the training works, how you get there, because if we really want to make scientific progress, we need a cohort of people coming through who are experts in both the science and the clinical work. So, first 5 minutes, I'm going to tell you a little bit about my journey to becoming a hematologist who specializes in MPN. So my journey begins here. This handsome-looking, thoughtful chap. So how did we end up from 1991 going to medical school at St. Peter's College in Oxford? How did we end up to me being a fully fledged kind of researcher investigating myeloproliferative neoplasms? Well, it's a long journey. This is the thing, and we are in a bit of a problem at the moment in terms of the next generation of clinician scientists, because this long journey, to a certain extent, is putting people off. So I started off in St. Peter's in 1991, finished medical school in 1997, did some house jobs, these are kind of junior doctor jobs in Oxford. And it's really kind of by chance, really, that I happened to get rotated to hematology, and had some very formative experiences there looking after patients on the ward who were going through transplants and having a really tough time. And that's one of the reasons why I decided to do hematology, and I'll tell you a few others in the next slide. So then got a training number as a junior doctor to become a specialist, did a PhD at UCL, which was supported by a big medical research council, the university funding, and got my PhD in 2007. Along the way, there are a lot of exams. So they're thankfully behind me now, but physiology, the first degree, then a clinical degree, then a member of the research of physicians, College of Physicians, Part 1 and Part 2, did my PhD. And before I tell you about kind of the next steps, I just wanted to just reflect a little bit about why I did hematology, because I think trying to encourage lots of other people coming through to be clinician scientists is really important, and we'll make a lot of progress. So there is a bit of serendipity. You know, I just happened to get rotated to hematology and had those formative experiences. It's a very clinically rewarding job. You get a choice of a wide range of different specialities. There's a clear unmet need to improve things for patients. Inspirational mentors. And I would, of course, particularly highlight Claire. You know, we're really lucky in the UK to have such a kind of huge kind of figure in the field of myeloproliferative neoplasms. And she's so welcoming. For me, when I started off specializing in MPN, incredibly welcoming. And it's not always like that for leaders in the field. So thanks very much, Claire. There's lots of academic opportunities for folks in hematology. But for me, one of the most important things was actually personal reasons. So here I am. We've just been climbing a ridge in Scotland. That's my brother, who's a neurologist, clinician scientist at UCL. That's my father, who had leukemia. He sadly died last year. And that you really learn as a family the impact of blood cancers at a personal level, and it's a real personal motivation and something that I think is common across many colleagues, that they're passionate about making a difference. It's not just a job. So, where do we go from here? So, the point I want to make in this slide, without running through everything, is number one, it's a long journey, and number two, there are absolutely key roles for research charities in supporting the journey along the way. So, my first kind of big grant was a fellowship funded by a research charity, a Bennett Fellowship, now from Blood Cancer UK. I'm 51 now, so I'm still supported by a research charity. This is something that you may not appreciate. I'm funded by Cancer Research UK, so the work I do in the NHS, a lot of it, and the work I do in the university is largely funded by research charities. So it makes a huge impact on what we're able to do. More exams. So here we go, 2003, 2008, the FLC PATH exams. Finally, I get my certificate of completion of training. So that takes a long time, a long time, 18 years or so, and finish. So it's a long journey. And just last couple of slides on this before I get on to some bona fide scientific updates. So how did I end up focusing on myeloproliferative neoplasms? Now, that was really by chance. I came to Oxford to do some research, and it just happened that no one was particularly focused on MPN in Oxford. So I thought, looks like a pretty interesting disease to work on, and I started setting up the clinic there. And so that really was serendipity. For clinician scientists, just to give you a sense of how we work, so about 2/3 of my time is running a laboratory, carrying out laboratory-based research, but that really is so, and that's done at the Weatherall Institute here in Oxford, but that's incredibly closely linked with my clinical work, running clinical trials. I'm not going to talk about clinical trials today because other people will do so, and just running the NHS service. So just finally, some reflections on why I really, really genuinely love what I do. I think first and foremost, it gives you an incredible opportunity to translate research through to patient benefit. Science is definitely almost like a hobby. It's very addictive, and you get hooked on it, it just makes going to work and doing what we do enjoyable. You get to kind of really integrate clinical and laboratory research skills. It's really rewarding working with research funders and other stakeholders to really help shape the kind of research that we do. You get as an academic a certain degree of freedom, although my wife says it's freedom to work every weekend. But I feel I've got some freedom anyhow. And you know, it's wonderful working with young scientists and mentoring a team and trying to encourage the next generation to come through. Okay, enough about that. I'm going to tell you about some scientific updates now. So I put on this cartoon. This is the same cartoon that I would show to a kind of in a research meeting, just summarizing the kind of life history of the development of a myeloproliferative neoplasm. So these diseases start many, many, many years, almost certainly decades. We talked about this at the last meeting last year. Probably decades before people actually present with a myeloproliferative neoplasm. You get a mutation in a single cell that then gradually grows. And the point is, it only grows in a minority of patients. That process takes a long, long time. And it goes through this phase that I just want to touch on a little bit, this disease, or not really a disease, this phenomenon called clonal hematopoiesis, where you can find mutations, but patients essentially, or persons, have got normal blood counts. And then after another period of many years, that can develop into early myeloproliferative neoplasms. And again, that only happens in a minority of people. And then once MPN is established, thankfully, it's only a minority where this progresses to a more advanced phase. So one terminology you might have heard of is this chip. I don't know if anyone's heard of this chip, but if you hear about the terminology chip, that is clonal hematopoiesis, this thing that I've just talked about, of indeterminate potential. So this has been recognized for a few years now. This is a situation where you have a person, typically an older person, with normal blood parameters, but you can find a mutation. And for the purposes of today, it's very frequent that you can find a mutation, for example, in JAK2 or calreticulin in people with completely normal blood counts. And the odds of that situation progressing to a more advanced form of MPN are actually very low. It's less, about 1 in 200 people per year where that mutation actually causes MPN. So understanding why that is really, really important. And the other really important thing is this creates a kind of window of opportunity to potentially even prevent people getting MPN in the first place. And I think that could be very important over the coming years. So can we kind of predict who is going to, in the rare situation, progress from this clonal hematopoiesis to another MPN? So this is a very, very large Danish study where they carried out very sensitive analysis for the JAK2 mutation, and to find it down to a very low level, 0.1%. And if you do that, you find 3% of the general population have got a detectable JAK2 mutation. The vast majority of those people have got normal blood count. Some have got abnormal blood count, but still the large majority do not. And if you measure the level of the mutation, the higher the level, we can assign this thing called an allele burden. It gives a percentage to the mutation. That correlates with an increased likelihood of having abnormal blood counts. And what this is kind of a very recent study from the group in Cambridge at the Sanger Institute, where they used data. You may have heard of the UK Biobank. This is an enormous national effort where about over 500,000 people have consented to take part in this enormous cohort study where they donate samples and are followed up and clinical information is collected. And as part of that study, they've had what's called exome sequencing carried out, looking for any mutations that are present in their genomes. And if you do that, you find a substantial proportion, as I was just saying, who have a JAK2 mutation. And what they did in this study is ask the question, does having a JAK2 mutation predict over the following years a chance that that person is going to be diagnosed with a myeloproliferative neoplasm? And that's shown in this plot here. So if the bar is over this side, that means it's an increased risk of developing, if you look at the red bars, a myeloproliferative neoplasm. So as you might expect, a JAK2 mutation and a calreticulin mutation found, remember, in people with normal blood parameters predicts for an increased risk of developing MPN over the coming years. And if you look, this is time in days, this is JAK2, this is CALR. These are people at the beginning, normal blood parameters, and then over time, over quite a few years, here's about a 10-year time point, you can see a proportion of these people are developing an MPN, and it very much depends on the level of the mutation. So they've developed a scoring system that you can use to if you pick up a mutation in someone with normal blood counts, you can score that and you can predict the risk of the disease progressing over time. And I think in many ways this is the way the field is going to go, particularly for MPN and similar diseases, that we'll be looking, can you potentially think of ways we can intervene earlier in the course of the disease to prevent things progressing from clonal hematopoiesis to MPN or from early MPN to a more advanced MPN. And that's what I want to talk about next. So, what are the recent research findings that can give us insights into how this process, thankfully a rare process, but in some patients this can happen where early MPN develops into a more advanced phase. And here I'll just give you a quote from a very, very famous molecular biologist called Sidney Brenner, which is, progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. So you think science is all about inspiration. Actually, a lot of scientific progress is about having amazing new technologies and techniques where we can study things. And the technique I want to mention today is a technique that my laboratory particularly focuses on, a technique called single-cell genomics, which is an incredibly powerful technique. And to kind of exemplify that, the classic analogy is the fruit smoothie. So if you take a blood sample and you make DNA from millions and millions of cells and essentially make a soup out of the cells, You can analyze that and you can find JAK2 mutations and similar mutations, but you lose an awful lot of the complexity. Whereas if you analyze each cell individually, you can pick out all of the potential individual components that have come together in our case and in our focus, to make an MPN progress from one phase to another. So we are putting quite an effort into studying this phase of the disease, the blast phase of myeloproliferative neoplasms. It is, thankfully, a very rare situation, but it does happen. And when it does happen, unfortunately, it is very, very difficult to treat. So we should be just thinking about how can we prevent it happening in the first place. We focused on this particular mutation called P53. This is a mutation that is, across all of cancers, is the most common mutation found in human cancer, but it is also found in myeloproliferative neoplasms and does predict for the disease progressing. And so we wanted to understand how the P53 mutations do that. So the important point here is, again, back to a kind of cartoon, is chronic phase MPN with JAK2 mutations. Many patients will have a P53 mutation, and thankfully, most of those patients never progress. So their disease never progresses to a more advanced phase. It only happens in a small number. So why is that? Why in some people do we see this progression, and in other people we do not? And we use this, all the different colors here represent different individual cells, trying to kind of convey this kind of genetic complexity. And you can imagine if you pick out all those different colors at the individual cell level, you can really start understanding what the mutation is doing. And we've used this technique, a single-cell genomic technique we developed in our lab called Target-seq to do that. One of the really powerful things we're able to do as clinician scientists is link up the kind of these cutting-edge laboratory techniques with collecting samples from clinical trials. So over a number of years, probably I think it must have started about 10 years ago, the PHASAR study. We ran a study combining JAK inhibitors with azacitidine treatment, so a novel combination treatment in people with more advanced forms of MPN. The important thing is we collected lots of samples from these patients. Patients, unfortunately, need to have a bone marrow. We collect blood samples, and many of you will have taken part in research studies, so I thought it would be interesting just to show you what we do with those research samples. We separate them out into different cellular compartments, so with the white blood cells, the platelets, the bone marrow, and store things down, and then we can analyze them using these cutting-edge techniques. Every sample we get coming through the lab, and we do a lot of this processing in the lab, takes about 4 hours, so it's a huge endeavor to collect these samples and store them. So in the PHASAR study, we did that, and we were interested particularly in these mutations. So each little block in this waterfall plot represents an individual person in a column that has an advanced form of MPN, and each one of these got a P53 mutation. So there were quite a few patients who had an advanced form of MPN and a P53 mutation. So that allowed us to really study at the single cell level how this P53 mutation is causing secondary leukemia after a chronic phase of MPN. And we studied tens of thousands of cells in order to do that. And what we learned is that the P53 mutation alone is not enough to cause the disease to progress. You need to lose, so that, remember, with genes, there are 2 copies of a gene that we inherit. So if you get a mutation, that's 1 copy that's damaged. But the other copy has to be lost. It either gets deleted or mutated. And in addition to that, every patient that we studied in this situation also gained what are these things called copy number alterations. That means the damage to the chromosomes elsewhere, unrelated to P53. So the important point is P53 not enough to cause disease progression. You need this kind of multi-hit state where you lose the wild-type allele and gain a lot of chromosome abnormalities. And you could use this single-cell genomics approach to then study, well, what is it about the cells that eventually drive this disease progression that's so difficult to treat? And when we did that, we were able to learn that there's very, very strong inflammation. So inflammation is really emerging, I think, as a crucial mediator that causes progression of these of myeloproliferative neoplasms and interacts with the mutations. So I'll not go through some of the details of this study, but what we concluded was that this inflammatory signaling is really important, both for suppressing the normal blood cell production and promoting these p53 mutations to grow more aggressively and become more genetically complex. So the next step was to then say, well, if these chromosome levels, so not at the individual gene level, but looking across a whole chromosome, if they are the important events that occur, then can we do something to tackle those, to target those? And this is some work that was led by a really talented PhD student in my lab who's also a clinician, so she's going to come through as the next generation interested in MPN. She's called Charlotte Brearley, and she studied this process, got a kind of ancient Greek name, chromothripsis, which is, this is what a chromosome looks like. And in some people, the chromosomes during the development of cancer can become, essentially, it shatters and then gets put together in a higgledy-piggledy way that isn't quite accurate. Myeloid diseases, like myeloproliferative neoplasms, we thought chromothripsis was a very rare event. But the problem is MPNs are rare. So in this huge study across thousands and thousands of different cancers, there were just tiny numbers of MPN, and that's why we need people specifically asking questions relating to MPN. So when we studied a cohort of MPN patients, or Charlotte did, these are all the chromosomes lined up, you know, 22 autosomal and then the sex chromosomes X and Y. These kind of bar plots above show whether you've got an extra copy or a deletion of a part of a chromosome. Using a very specialist technique, we were able to find chromosome 21 in some of these patients really stood out as an area not previously recognized or reported to be causing problems in MPN patients. And in a significant proportion, it was this chromothripsis that was causing the problem. And in patients that had this chromothripsis event, unfortunately, it was a really, really difficult disease to treat. Conventional treatments did not work. And what Charlotte did, I'm just summarizing her work very briefly, was she identified that there was a particular gene on chromosome 21 that as a result of this chromothripsis becomes amplified. Some too many copies, instead of 2 copies, the patients had 8 copies of this gene. And the important thing there is, this is a gene where there's an inhibitor treatment, it's a tablet treatment that had been developed by drug companies for a completely different reason, actually as a diabetes treatment. That drug is available, and when we tested it in the laboratory, we found that patient samples with this abnormal chromosome 21 were very, very susceptible to inhibition with that drug treatment. And it just shows how by collecting samples, running clinical trials, really studying them carefully, you can come up with potentially new treatments where other treatments, standard treatments, are failing. Okay, I've just got 2 or 3 more minutes now, and hopefully there'll be a bit of time if you've got any questions. So one of the kind of really exciting technologies, again, if we go back to technologies in the field, is artificial intelligence. And that is now being applied for MPN diagnostics. So a Spanish group have developed this artificial intelligence approach to risk stratify persons with myeloproliferative neoplasms. This is the standard prognostic scoring system, and then using artificial intelligence, you could see that we were able to reclassify much more effectively. And I think this kind of technology is coming and will get gradually incorporated into how we diagnose and more precisely convey the potential impact of a disease for patients. My colleague Daniel Royston in Oxford is using a similar approach to study bone marrow biopsies. The way we study bone marrow fibrosis it's been the same for many decades. There's a As many of you will know, there's a scoring system where you assign scarring in the marrow as 0, 1, 2, or 3. And Dan has developed an artificial intelligence approach to much more precisely quantify that fibrosis. So this is now starting to, not in routine clinical practice, but the samples coming through the lab, he's really trying to incorporate this type of approach more routinely to more precisely predict whether someone with MPN is at risk of developing fibrosis down the line. Another thing that we're doing in Oxford, we're running a study that should be open next year where we're going to carry out some imaging, so MRI-based approaches, with the idea, we hope, that instead of needing a bone marrow biopsy to test for whether someone has fibrosis, you can actually do that with an MRI scan that I think anyone that's had a bone marrow would agree would be beneficial. Yeah, I can. Don't need any convincing of that. The MAGIC study, just in the final slide, I just want to mention, this was published earlier this year, an absolutely crucial study. I know people in the room have taken part in this study and are making absolutely crucial advances in the field for myeloproliferative neoplasm, showing that ruxolitinib can really suppress the mutation level in persons with polycythemia vera. And the suppression of the mutation level, if we achieve it, really correlates with markedly improved outcomes for patients. And this also applies, by the way, after transplant. So if the mutation levels are suppressed, the outcomes are much better. So I'm going to leave it there, just to say that, what I've summarized is most patients, probably all patients with MPN, will develop the disease after a period of clonal hematopoiesis. This provides a window of opportunity to intervene, to potentially prevent the disease. We're learning much more about this process with single-cell genomics. Inflammation looks like it has a really central role. So one of the questions is, can you use anti-inflammatory treatments to potentially prevent the disease progression? And genetic abnormalities at the chromosome, rather than just the gene level, can potentially be therapeutically targetable. So thank you very much.